and welcome to this help video. I understand that some people are having difficulty with the SNP lab assignment and people have asked for help. That's why I'm making this video. Okay, so let's start on Canvas, and we're in Unit 4 here, and you can see at the bottom of Unit 4 is the SNP lab. So let's go ahead and click on that and get started. All right, it opens up the SNP lab in Canvas, and what you'll see is a lot of background information. There's a lot of text here that tells you what SNPs are, what they're used for, and there's also this lecture video, again, with background on SNPs. I'm not going to cover all of that. That's there for you to access as you please and when you can. What I'm going to concentrate on is actually getting you through the activity. All right, I'm going to walk through the performance of this lab. And I'm going to do that by clicking right here, which takes us to the Google Doc. That is this lab. So this is a Google Doc, and of course your assignment is to fill out and submit the Google Doc. Now whenever dealing with Google Docs, the first thing we have to do is make a copy. Because you cannot edit the Google Doc that you access from Canvas. That's my Google Doc or the class copy, and you don't have edit privileges. So you have to make a copy, and as soon as it comes up, now you have a version that you can edit. Okay, so there it is. We're ready to go. All right, and the first thing it tells us to do is to go to SNPpedia. All right, so let's go ahead and do that. All right, here's the SNPpedia homepage, and you can see it's got a little information up at the top, what SNPpedia is and why you might want to use it. It's got some help screens and help topics. If you need more information on anything, go ahead and go to those. And then it has this section that says popular. So these are SNPs that have been in the news lately. All right, they're of interest in the recent past. All right, and you can see there's some interesting things here. For example, here's a SNP that's associated with social behavior, a SNP that's associated with muscle performance or with Alzheimer's. All right, and this one supposedly has a big effect on whether or not you can get Alzheimer's disease. So a lot of interesting stuff here. Go ahead and feel free to access it and use it as part of the assignment. But I'm going to do something a little bit different. I'm going to search for something that I'm interested in. Now, I know that caffeine has been in the news lately because we're beginning to discover the genetic basis for differences in sensitivity to caffeine. I myself suspect that I am very sensitive to caffeine. So this is a topic that I'm very interested in. All right, so if I click caffeine in Wikipedia, excuse me, I'm in SNPpedia, and it tells me a little bit about caffeine, about the compound, about the drug, and there's even a link here to Wikipedia if you want to learn more. But what I'm interested in is the SNPs. And this does not necessarily list many SNPs. What it mostly is links to genes, links to published articles that include some SNPs and some SNP associations. But there are a couple SNPs right up at the top here. All right, and this one refers to a scientific journal, and what that article says is that these SNPs here influence habitual caffeine consumption. All right, so that's kind of along the lines of what I'm interested in. So I'm going to go ahead and grab this SNP. And part of the exercise is for you to include these SNP names. So I'm going to go ahead and capture that SNP name. Another part of the exercise is for you to include some articles. And here's a link to an article which may be of interest to us. All right, but let's take that SNP and paste it right in here. And what I'm going to do is I'm going to paste it as plain text, which on a Mac is Shift Option Command V. Okay, plain text as opposed to formatted text. Now it's asking us for 3 to 5 SNPs. You can go ahead and do that. I'm just going to work with 2 for now. It's also asking us why we're interested in these SNPs. So let me go ahead and write a brief description. All right, that looks like a pretty good explanation. Now I'm going to look for another one. So I'm going to go back to SNPpedia, and I don't want to lose this information. I want to keep this page, so I'm just going to open up a new tab with in SNPpedia, and I'm going to do another search. This time, something that we're probably all interested in, which is obesity. And when I do this search, you'll see that there's a whole bunch of SNPs. These are all SNPs. Now, in this case, all of these SNPs are associated with genes, okay? So this is a gene, this is a gene, these are SNPs. The RS numbers are SNPs, the capital letters are genes. And there's a lot of them here, and there's actually a lot more than is listed here. I'm going to focus in on just one of those, and that's this one here. Again, the gene in this case is MC4R. MC4R stands for melanocortin receptor number 4. Melanocortin is a neurotransmitter. It acts in the brain, and it acts on appetite centers in the hypothalamus. It actually suppresses appetite. So the receptor for melanocortin is called melanocortin receptor. And as you can see, there's more than one of them. There's melanocortin receptor 1, 2, 3, and 4. All right, so this is the SNP, but this SNP goes by another name, V103I. And it has been associated with appetite regulation, okay? And as such, it affects BMI. BMI is a measure of obesity. Okay, so this seems to fall in the area that I want to investigate. Now let me just refer to this other name for a moment, V103I. What that means, what that refers to, is an amino acid substitution. All right, so in other words, there is normally a valine at position 103. Okay, so that's V103, valine at position 103. In the case of the SNP, or in the case of the minor allele, that valine is substituted with isoleucine. All right, so valine 103 isoleucine. All right, so all SNPs that lead to a coding sequence change can be represented in this way. But once again, I'm going to grab that SNP because that's part of our assignment. And let me also paste that one into our Google Doc and give a little explanation for it. All right, so there's a couple of SNPs. Again, your instructions are to look for 3 to 5 different SNPs. I'm going to stop here at 2, but you're going to carry 2 through the rest of this. I'm actually going to carry forward just 1, the top one here. Okay, our next step is to go to the UCSC genome browser. UCSC, of course, stands for University of California, Santa Cruz. So their bioinformatics department has been a world leader in generating, organizing, and displaying lots of genomic data from many different sources. And their genome browser is now quite famous, and it's used by scientists all over the world. All right, so let's go there. Okay, so this is the front page, and the instructions tell you to go to the genome browser, and that's the top link right here. All right, and then from here, again, there's many things we can do, but one of the things we can do is to perform a search. So I'm going to go ahead and search that caffeine SNP, and you'll see that I get several hits, many hits actually. All right, so what this says is catalog of published genome-wide association studies, genome-wide association study, or GWAS. So that means that this particular SNP has come up in many different GWAS studies. But it's the same SNP in every case, okay? So this shows each one of these studies has shown an association between this SNP and something. And I think most of these are related to caffeine metabolism. Once again, they're all the same, and you can look very closely at the sequence that it's referring to, and you'll see that it's all the same. But these are big numbers, all right? So let's just concentrate on the last 4 digits, 6858 to 7358. In other words, this is a 500 base pair region of the genome, and these are all the same. Now this one, same region, but it's just a 200 base pair region of the genome. Let's start there. That's going to be a little bit simpler. All right, so now this is the genome browser proper, and what you see, this vertical yellow line in the middle represents our SNP. All right, and here it is categorized RS2470893. And the first thing we want to do is paste into our Google Doc where it is, okay? And this tells us exactly where it is. Chromosome 15, nucleotides, 74,727,008 to 74,727,208. Okay, so a 201 base pair window. Let's go ahead and copy that information. And once again, paste it into our Google Doc. All right, so this is asking where is that SNP? And the answer to that question is right here. Now that's a range of 200 base pairs, 201 base pairs. But you can narrow it down to a single nucleotide by zooming in on that genome browser. But we don't have to worry about it in such fine detail right now. And now it wants to know the actual nucleotide variation. Okay, so write out the nucleotide variation. And again, you're going to be carrying 2 SNPs forward. I'm working on just 1. All right, so let's go back to our genome browser. And it's right here, actually, this is the information right here, RS2470893, that's our SNP, C to T. Okay, in other words, the major allele is C and the minor allele is T. Now, that is, most people will have the nucleotide C at that position, but a minority of people will have a T. All right, so that's really the information we need. Let's go ahead and copy that down. Alrighty, so that's one way to represent the actual nucleotide variation. Now, along with this Google Sheet, there is this graphic, which is an annotated genome browser window telling you where everything is. I'm going through this in real time, so we don't really need to look at that. Hopefully you're going to gain that as you go through this video. Okay, now the next part is a little bit tricky. It says to identify the nearest gene. All right, so let's go back to our genome browser. And you'll see there are no genes. If there were genes, they would be showing up right here, ref-seq genes, all right? But there are none. And the reason there are none is because we're focused on too narrow a region of the human genome. We're looking at just a 201 base pair region. That's not unlikely to show any genes. So what we really have to do is zoom out. And I'm going to take some big steps out. There's 1 step out. There's 2 steps out. And now we're looking at a window that should be about 20,000 base pairs. All right, and now we can begin to see genes. So this is a representation of a gene in our genome browser. All right, so you do have to really take a step back and look at a big chunk of genome in order to start seeing genes. Because remember, most genes will span thousands of base pairs. All right, so this is how the gene is represented in the genome browser. It's represented in blue. It's got a thin line and it's got some thick lines. Okay, so those thick lines are exons. All right, so for example, if I mouse over there or mouse over there, it will tell me that that is exon 1 of 7. The thin line is intron. And if I mouse over there, it says that's intron 1 of 6. All right, so this gene has 7 exons. And if it has 7 exons, it has 6 introns. All right, now if we look at the thick lines that represent the thick lines that represent exons, we'll see they come in 2 different sizes. There's this thinner one and the thicker one. Thicker one and thinner one. All right, so the thicker ones represent coding sequence. All right, so the exons are all going to wind up in the mature mRNA. The introns get spliced out. But within that mature mRNA, there's the coding sequence, that is from start codon to stop codon. But there's also sequence that's upstream and downstream of that. 5' untranslated region and 3' untranslated region. All right, so these thinner blue lines there and a little bit of it right there represent 5' UTR. And then over here, this thinner line represents 3' UTR. All right, and note that there are arrowheads. Okay, so this is showing that this gene runs from right to left. All right, so the 5' end is here, the 3' end is over here. All right, so it's asking us for the ref-seq number, okay? And the ref-seq number is right there. It's that NM number. Actually, before it asks us for the ref-seq number, I got a little ahead of myself. It's asking us for the gene name. Okay, so the gene name is right there, CYP1A1. All right, so that's the name of the gene. Let's go ahead and write that down. Okay, so to get to the gene, we had to zoom out, and once we zoom out enough, we should be able to see one or more nearby genes. And this gene is CYP1A1. All right, so CYP stands for cytochrome P450. Cytochrome P450 is a superfamily of genes. That means that it's a very large family with many different branches. It's a superfamily of genes that encode P450 enzymes. P450 enzymes are membrane-embedded redox enzymes. They generally carry out oxidations. All right, and you're familiar with one such enzyme. Cytochrome C falls into this family. Cytochrome C is in the inner mitochondrial membrane, and it plays a role in electron transport, a redox role in electron transport. All right, all the CYP450s are membrane embedded. Some of them are in the mitochondria, some of them are in ER, some of them are in peroxisomes. Okay, CYP1 is a subfamily within that superfamily, and then A represents another subbranch, and then 1 represents one gene. All right, so it's a large family with many different branches, CYP1A1. All right, it's a large family of genes, so the nomenclature gets a little bit tricky. So these P450 enzymes, like I said, carry out oxidation, and that's part of a larger strategy that humans and a lot of animals have for detoxifying foreign compounds. Okay, so they play a role in detoxifying and getting rid of foreign compounds, including drugs. And and caffeine is one such drug. The general strategy for getting rid of foreign compounds is not to see if they're useful at all, but just to get rid of them. And the way you do that is to oxidize them. Oxidize them and make them more water soluble. For example, when you oxidize, you make things more water soluble. For example, hydrocarbons can be oxidized partially to glucose, and of course glucose is much more water soluble than hydrocarbon. So once they're made water soluble, they can be excreted in the urine. Okay, so that's the general idea, and this happens a lot in the liver, but it can happen in other tissues as well. In the case of caffeine, it does get oxidized and excreted, but of course it also has actions in the brain as a stimulant. All right, so let me go back to the genome browser, and I want to show you something else before we go on. And to do that, I'm going to zoom out one more time. All right, and if we see, once again, this is the gene we saw before, CYP1A1, and here's another gene, CYP1A2. And as you can guess, it's also a cytochrome P450. In fact, it's a very closely related cytochrome P450. Same subfamily, same group, but a different gene. All right, so these are very closely related. Now here's a situation where we have two closely related genes near one another on the same chromosome. Okay, so your brain should light up at that point. So now you should be thinking unequal crossover, right? Because we learned earlier that unequal crossover can lead to gene duplication. And that's essentially what we have here. The gene gets duplicated, then they each evolve on their own to become slightly different genes with slightly different functions or slightly different substrate specificities or regulatory modes or whatever. Now, there's a little bit more going on here because you can see that these things are in opposite orientation of one another. Okay, so CYP1A2 runs left to right, 5' to 3', and CYP1A1, 5' to 3', is right to left. So there's an inversion going on as well. Okay, so with that aside, the next thing we want to do is come up with the RefSeq identifier. Okay, so RefSeq is a reference database of all of the sequence of all genes, or really of all mRNAs. All right, and it asks us to look for that using the nucleotide browser. So this says nucleotide search. And we can go ahead and do it that way. And I'll just give you a quick example. So if I write in here CYP1A1, there it is right there, the Homo sapiens gene for cytochrome P450 1A1. All right, but we don't need to do that. So, and that sequence will be available there. But we don't necessarily need to do that because these links are all live. They're not live right now, so something funny happens when I click around. If I click there and I get all that extra information that I don't need, but now now this becomes clickable. So I can go ahead and click on that refseq number. Okay, and so there it is again, the refseq number. And if I click on this, so I'm still in the UCSC genome browser, but if I click here, it takes me to NCBI. NCBI is the National Center for Biotechnology Information. It's written right there. And this shows me information about this particular gene, once again, CYP1A1. And it's a lot of different information. If I scroll all the way to the bottom, I'll get the nucleotide sequence of that mRNA. Yes, this is mRNA sequence, even though it doesn't look like it. It is. But there's a lot of other information too. Okay, so most of this is references that tell you, that really reference the papers that originally sequenced this gene. All right, so there's a lot of references here. And a lot of the linkage data is here, so all kinds of references. Then a quick summary down here. And then we get features of the mRNA. Okay, so these are features. For example, this mRNA is 2,603 base pairs long. All right, it tells us where certain exons are. But it also gives us the coding sequence. Coding sequence is abbreviated CDS. And what it's saying is that the coding sequence is from 118 to 1656. That is, the ATG is at 118, 19, and 20. And then the stop codon is at 1654, 5, and 6. All right, so this is the whole coding sequence. And remember, we also have 5' UTR and 3' UTR. All right, so here's that coding sequence, and they've actually taken the trouble and translated it for you. So this is amino acid sequence. You may not be accustomed to seeing amino acids as single letter abbreviations, but each amino acid, in addition to having a 3-letter abbreviation, also has a single letter abbreviation. And that's what we're seeing here. So I can go ahead and copy and paste this into Google Doc to answer the question, but I'm going to do something a little bit different. I'm going to click on this link, which is the NP number. NM, the M is for NM, the N is for mRNA, but NP, the P is for protein. So I'm going to go ahead and click on that link. And once again, I'm getting to a page with a lot of information, and once again at the very bottom is that amino acid sequence. But I'm going to display this in a simpler form, a shorter, simpler form, which is by clicking on FASTA. And there I have the amino acid sequence, plus a very brief descriptor of what that sequence is. So I'm going to go ahead and copy that sequence. And I'm going to paste it into our Google Doc. All right, it says replace this text with the amino acid sequence. I'm missing a word there, but you know what it means. And in this case, I'm going to paste it as formatted text. All right, and I skipped this one. It says replace this text with the refseq identifier number. So I can go back and get that. Again, back to our genome browser, and that's the actually I can take that. I can take it from here too. I can take it from this page. Here it is right up at the top. Nope, not there. I can take it from this page, and here it is right at the top, NM. All right, so I think that answers that question. All right, and once again, you'll be doing that with a second SNP as well. All right, now number 6 is a question, kind of an interesting question. Based on the nucleotide alignment of your SNP sequences among vertebrates, do you think that using a mouse model would be beneficial to gain further information about each of these SNPs? Why or why not? All right, so to answer that question, let's go back to the genome browser. And this, I need to go back one more page from here. So I'm just hitting the back button. All right, and here we are back at our genome browser. And here we are once again in the SNP, that vertical yellow line. Now if I scroll down, or if I look down on the page, I come to this section, and this is the conservation among many vertebrate species. It says alignment among 100 vertebrates. Okay, so Humans are vertebrates, as are rhesus monkeys and these other mammals, as well as birds, amphibians, and fish. And what this is saying is that each black line represents conservation of a nucleotide between human and whatever of these species is represented on each line. And of course, those black lines, those black vertical lines, can get very compressed because remember, we're looking at 60,000 base pairs of sequence in this window. All right, and what this is telling us is that if we look at rhesus monkey, we see that there's a lot of conservation, not only in the gene region, okay, so there's the gene, but also in these intergenic regions. There's no genes here, but there's still a lot of conservation. And then we come to the next gene, and once again, a lot of conservation. Okay, that's not that surprising because the monkey is a primate and relatively closely related to human. If we go to something a little bit further away, like the mouse, which is the second line, we'll see that we still retain this conservation in the area close to gene, but in the conservation in the intergenic regions has more or less disappeared. It's back a little bit when we come to the next gene, but so there is conservation, but it's much less. And then if we go to more distant species like bird, amphibian, or fish, we'll see that the conservation really only happens in the regions that are close to a gene. Okay, all this intergenic conservation has disappeared. And actually, even within the gene, it's only the coding sequence that's conserved. The introns are not conserved, and some of the 3' and 5' UTR is not conserved. All right, so the further we go, the less conservation we see, but that conservation, of course, is concentrated in coding sequence because in order to have any activity at all, the gene has to include conserved coding sequence so it carries out enzymatic activity. All right, but the question is specifically about mouse, okay, and it's about this SNP in mouse. Now note that this SNP is upstream of the gene, okay? So this is the 5' end of the gene. The SNP is upstream of that gene. All right, so therefore it does not affect the coding sequence, all right? There's no change in the structure of the protein caused by this SNP. It is, however, in an upstream region, and we know that upstream regions can play a role in regulating the expression of a gene. And not only that, but in this particular upstream region, there seems to be conservation between mouse and human, at least some conservation. All right, so it's still a difficult question to answer, but I'm going to try to answer it as best I can. All right, so that's a pretty good answer. Once again, you're taking 2 things forward. All right, so the next question is to list published genome-wide association studies for your SNP. And to do that, we're going to go back to SNPpedia. Now, there's a link here for the proper format for references. All right, and I'm not going to click on that. You can click on it if you want, but I'm going to show you something that's a little bit simpler than going by that reference. Then going by that particular style. All right, so to get this answer, we're going to go back to SNPpedia. Here we are, SNPpedia, and once again, this is the SNP that we've been following. So let's click on that, and here you see a whole bunch of GWAS references. Okay, so here we go. So this one has to do with caffeine, so we can look at that one. But I'm going to look at this one. It's a little bit easier to handle to some extent. Okay, well, let's take a look at this one. Okay, so once again, habitual caffeine consumption. All right, so I'll click on that, and this takes us once again to the National Center for Biotechnology Information. In addition to housing all of that genomic data, they also house the National Library of Medicine, which is links to articles that are published in thousands of different biomedical journals on a monthly basis. All right, and this is part of the National Institutes of Health, which of course is part of the federal government. All right, so the federal government has really done a lot in terms of advancing genomic research. Okay, so here is the article, Genome-Wide Meta-Analysis Identifies 6 Novel Loci Associated with Habitual Coffee Consumption. So a meta-analysis combines the results of many different individual genome-wide association studies in order to get more statistical power and to find associations that may have been missed initially. And so if you go down, there's an abstract, which is a quick summary of the paper, but there is this long list of authors. All right, so that's a very long list of authors. You often see that in GWAS papers because it's a lot of work. I mean, we're talking about thousands of individuals being genotyped for tens of thousands or hundreds of thousands of individual SNPs. All right, so that's a lot of data, millions of data points, tens of millions of data points. And so all that work has to be done, and of course all that statistical analysis has to be done. So there's a lot of people who go into this. But the quick name of all these authors is right here, the Coffee and Caffeine Genetics Consortium. So I'm just going to stick to that for the author. Now, instead of copying all this, there is a quick way to get this reference, and that's right here. It says cite. And so here we have the reference to that paper. And once again, it includes that long list of authors, which I'm going to delete, but the rest of the reference information is here as well. So I'm going to go ahead and copy that. And now I'm going back to my Google Doc, and I'm going to paste it in here. And once again, I'm going to paste it as plain text. And there it is with that long list of authors. But like I said, I'm going to omit all of these authors and just go with the Coffee Consortium. So that means all the way up to here. Okay, so now we have it in the proper format. Okay, so author, again, not real author names, but the Coffee and Caffeine Consortium. And then the title, the journal, Molecular Psychiatry, the year, 2015, and then the particular volume, which contains this article, and the page numbers. All right, and then there's additional information here, which is not part of the proper format, but that's okay. We'll just leave it there. Okay, so that's one reference. Of course, you'll have to come up with some more as well. That brings us to the next question. Based on the information you've been able to collect, do you think your SNP is causative or non-causative? Explain. All right, so all the information that we have so far tells us that this SNP is associated with the caffeine metabolism phenotype, with some differences in caffeine metabolism. Okay, but association is not causation. Is it causally involved? That's always difficult to answer. Most of the time, the answer is no. But you never know in any particular situation. What I've already stated is that this SNP is not in coding sequence. It's in it's in upstream sequence, and therefore I think it's not causally related. It may, and even to change the regulation of a gene generally takes a lot of sequence variation, not just one nucleotide difference. So I think it's a non-causative SNP. All right, so that's one answer. Again, whatever you answer, you have to come up with an explanation for what you say. All right, then the last part is to take screenshots, so we can go ahead and do that. Let's first take a screenshot of the genomic region. All right, it says the genomic region including the multiple alignments, and then the GWAS. I don't want that. Let's go back here to the genome browser. Okay, so here's our gene, here's our SNP, here's our region, here are the alignments. So let me try to capture all of that. So on a Mac, to do a screenshot, what you do is Shift Command 4, and there you see the crosshairs. So like I said, I'm going to try to capture all of this information. And that looks pretty good, about that much. All right, so let me open that up. Now I also want to take a screenshot of the SNP of the GWAS. All right, here's one of those GWAS. Let me go back. Here are the others. Once again, screen capture. Okay, that all looks pretty good for one SNP. Okay, so like I said, I carried one SNP forward. You have to go through the whole thing with two SNPs. I hope this helps. I think you can do it now. But if you need more help, please send an email. And good luck. See you later.